Welcome, everyone. Today, we're going to talk about ideas labs, synthetic biology, and building synthetic neurons. My name is Costa Michaelides. I'm a facilitator with Knowinnovation, which is K-N-O-W innovation. It works better written down. That's our running joke anyway. And I am joined today by University of Michigan professor and synthetic biologist, Alan Liu. Alan, welcome.
Oh, thank you. Thank you, Costa. It was very nice to see you.
Absolutely. Good to see you again. Okay, so let's go back to February 2018 and talk about the National Science Foundation's SynCell Ideas Lab. For those that don't know, an Ideas Lab is a 5-day conference for an interdisciplinary group of 30 scientists or so working to generate innovative research projects under a visionary theme. So in our case, at SynCell in 2018, the theme was building synthetic cells. Alan was one of our 30 resident scientists for the lab. Might have been 2019. Could be off by a year.
It was 2019, yes.
It was 2019, yes.
Yeah.
Okay. It was February 2019, if I remember the month correctly. All right. So Alan was one of our resident scientists. Alan, what was your experience at the lab?
Yeah, so this was definitely a very phenomenal experience for me, unlike any meetings I've ever been to. So just put things into context, at the time I was actually taking my sabbatical leave and spending a year in Germany on a humble fellowship. So I actually flew back to the US just for the Ideas Lab for 1 week. As you mentioned, there were about 30 PIs, 35 PIs from a variety of backgrounds from across many institutions in the US. So it was advertised as an intensive workshop focused on finding innovative solutions to grand challenge problems. So it was about synthetic cells, and surely it was very, very intense. I think coupled with my jet lag and the amount of discussion and work that we had to do at a time, I think I had about 4 hours of sleep on average a day for the whole week.
But that sounds about right for an Ideas Lab.
It was crazy. It was definitely crazy. But it was a lot of fun, and I met many great colleagues. The workshop took us to many exercises to explore big ideas and refine them. I think a lot of the workshop was geared towards creating teams and going through iterative feedback and on the proposed idea with all the participants there. So I had a blast. It was amazing.
Very cool. All right. So one important thing about Ideas Labs is that they're interdisciplinary. So this becomes important when you're tackling certain types of challenges. You just can't do it with a single discipline. You have to start sort of combining powers from across disciplinary boundaries. There's a lot to say on this topic. The NSF talks about convergence. It's evident that resolving some of the biggest mysteries in science requires an interdisciplinary approach. What's your experience been with regards to interdisciplinarity, just in general? And where do you think the scientific community is headed in that dimension?
Sure. Yeah, this is a great question. So I think this term convergence research was actually identified at NSF as one of the 10 big ideas for future NSF investment. So let me just speak a little bit about convergence research. I think it's a means to solve complex problems focusing on societal needs. And it integrates knowledge, methods, expertise from, like you said, different disciplines, and forming novel frameworks to catalyze scientific discovery and innovation. And in some way, you mentioned interdisciplinarity, but it spans across multiple disciplines. So the terms that are used within, sort of academia are transdisciplinarity, interdisciplinarity, and multidisciplinarity. So I think the closest one to convergence research is actually transdisciplinarity. So I'm a big fan of interdisciplinary research. My own training experience actually speak to this belief. Just to give you a little bit of a scientific background, I actually did my undergraduate degree in biochemistry, went on to do my PhD in biophysics and a postdoctoral research in cell biology, and up in the engineering school. So I think so my own training experience actually crosses multiple disciplines, and I'm a big fan of interdisciplinary research. I think my current group also reflects that. I have graduate students from mechanical engineering, biomedical engineering, chemical engineering, applied physics, cell and molecular biology, and biophysics. So my group is well represented across multiple department on campus. Let me say one more thing about I think your last question is about what I think in general scientific community is heading in this convergence research. I think there are two characteristics that defines convergent research, and one is research that are driven by a specific and compelling problem, So we're talking about deep scientific questions or pressing societal needs. And the second is, a deep integration across disciplines, right? So, bring together experts from different disciplines to pursue common research challenges, and that would utilize knowledge, theories, methods, data, across different communities. So, I think both of these, characteristics are very much still in play in today's, research, as well as, going to the future. That we do need to have a very, for certain problems, let's say climate change, for instance, it's a pressing societal needs, or certain biomedical or medical challenges that needs to be a sort of focused problem, and very specific problem, and then other questions require integration across the disciplines.
Yeah, it's interesting. And then I'm thinking back to, so I've been doing workshops and conferences with Knowinnovation since 2012 or so. And I remember going to my first academic conference. I'm not a scientist, just for clarity for everyone. I know
Yeah, I
Alan
remember
knows.
that.
We facilitate scientific
Like
conferences. Yeah, but our expertise is not science. We've become very science literate over time, which is really one of my favorite parts of the job. But I had not been to a proper academic conference until 2 years into doing the kind of work that we do. So our work's dynamic, immersive. Everyone's doing something. The walls are covered in post-it notes and flip charts. By the end, it's just very dynamic. Sometimes we have 16-hour days. It gets really intense. So I went to my first proper academic conference. We were hired to film it or record it. So we put up cameras in the back of the room to broadcast the conference. And it was like keynote Q&A, panel Q&A.
Right.
And then that was broken up by, meal time, coffee time, and networking. And it just struck me that you could have emailed the whole conference. Like there was no reason to fly everyone to, in this case, the Arctic. It was the Arctic Circle Conference. It's like, wow. Yeah. So the differences are really profound and interesting.
Yeah, and I think one of the things about conferences actually, not just in the talks themselves, as you said, they were basically tossing Q&A, but it's the hallway conversations, right? So sort of like you get out of the talks, you run to colleagues, and you start talking to them, and that's where, interesting discussion, take place, right?
Yeah, absolutely. All right, so creativity is another element of Ideas Labs, and it's one that we give great care to on our team. How has creativity played a role in the work that you do?
Oh, it's a great question. Yeah, so I think creativity is something that I value a lot in the scientific process. I have to sort of admit that this is one of the aspects about being a scientist that I like the most about, is being able to think about crazy ideas and really immerse myself into that process. So I think in some way the Ideas Lab was perfect for someone like me who really likes to challenge the way we think about problems, right? So I think it has a lot of brainstorming activities, both individually and in small groups, that really help in creating ideas. And so at the Ideas Lab, if I recall, we're encouraged not to jump to judgment about an idea. Instead, we're encouraged to come up with as many why ideas as possible, right? So one of the, you mentioned Post-it earlier. I remember having to write many things on the Post-it. It was almost like a mini competition of how many ideas you can come up with in the shortest amount of time. I think in that sort of scenario, each segment that in this Ideas Lab have focused questions. So we actually get to focus on a topic, and the team that we ended up with, having brought together interesting ideas and made the process more fun. So now talking about creativity more generally, when I was doing my PhD and postdoc, I really enjoy coming up with creative ideas. So if I think back my own independent work, I generally don't like to do things that other people have worked on. So I get the most excited when I'm working on things that no one else is working on. It's kind of an odd way of thinking about it. Is like, if someone else is working on it, I prefer not to do that work, right? So oftentimes this involves taking ideas, concept approaches in different fields, right? The way I go through this creative process is to basically extract ideas from other areas and applying to my own area. And as a PI now, I think creativity is one of the hardest skills to teach, but I think it can be fostered, right? So the activities that we did at the Ideas Lab was really great in bringing creativity out of individuals. So in my own lab now, I just try to encourage my trainees as much as possible to think outside the box and to try things that are outside of what we have discussed. And occasionally, there have been times where my trainee come up with really creative ideas, and that takes the science in the new directions, right?
Really cool. One of the ways I think about it, and I think this work was originally done by Teresa Amabile at Harvard, if I'm not mistaken. And so we look at domain skills. So if you're trying to be innovative, if you're trying to make a breakthrough in something, there's there are three sort of overlapping circles. One is domain skills. Like if you're going to be innovative in fashion, you have to know a thing or two about fashion. Right? If you're going to innovate something in an engineering field, you have to be in that engineering field. You have to know that field. Yeah. But what's interesting is that's not enough. So just because you know that field doesn't mean you're going to be innovative there. Another element is these creativity skills, the ability to generate lots of ideas, ask the right questions, really dig in, think laterally. I mean, the terms are numerous and the skills are numerous, but that's that creativity domain. That's the set of skills that allows you to come up with original thoughts. Yeah. And then the third piece is motivational, which is a whole other interesting sort of road to go down. But if you have the motivation, the domain skills, and the creative skills, you can make it happen. I did want to ask, so in your career, when did the creativity come into it? Were you always sort of creative in the work that you do as you became a scientist, or did you have to? Mm-hmm.
No, this is great. I think a lot about sort of how certain soft skills are developed. I think oftentimes it's not like there's a switch. I feel like sort of think about my interest in science. It obviously started when I was very young. Being exploratory, being interested in nature and go out and ask questions. And I think that is part of what fostered me into the kind of scientist I'm today. And I think there's not like a training that you go through to become creative. So when I discuss ideas with my group, sometimes you can see certain students would take conversation in directions where we didn't start out this way, but we ended up in a different place. And I think that is a good testament to being creative, because you're able to lead things into different directions and thinking outside of what we are initially start out with. In my own case, I feel like starting in graduate school, I was given a project, I started working on it, and I just started taking ideas from different places and started thinking, what can I do with this? And one can say this is a little bit defocused, but I enjoy the process of figuring out things on my own, and also just try to discuss with other people about potentially what my study could influence other fields. So it's hard to articulate exactly how these creativity come from, or how to build on that. It's still something I'm trying to do constantly, right? When I write a proposal, I have to come up with a new idea, right? So how do you do that? And people ask me, how do you come up with a proposal idea? So, well, you just look around and think, and something comes out, right? So I guess it's a similar process to that, right?
It definitely comes in different styles, and with scientists, you find a lot of puzzle solvers, so most of us understand puzzles. Yep. We've encountered one or two. You might hate them, you might love them. I'm not trying to make a judgment call, but you understand the process of like, there's this thing, I'm trying to untie these two little pieces of metal. And then you go through a process of either asking questions about the nature, like, why isn't this space big enough for the metal to fit through it? Or you either are asking questions, or you kind of sit there and just try lots of different things. What if I put it here?
You figure out the rules by doing that.
Yeah, you're kind of bending the rules. Mm-hmm.
Yeah.
So there's the question asking and the sort of trying lots of different things aspect. So that's the puzzle solver. And then there's all these other styles and ways of doing it. But it's really interesting to see them come up at conferences and to see different people's styles, intermingle. Definitely. All right. So tell us about synthetic biology and building synthetic cells and building a synthetic neuron, which was your group's project.
Great. Okay, so you hit on several questions there, and this is obviously a topic that's dear to my heart. So I'll start with the first one being synthetic biology, and sort of to give a sense of what that is and where a synthetic cell fits in. So as you may already know, that synthetic biology is a field of science that involves redesigning organisms for useful purposes. So it's a somewhat of an engineering discipline by engineering them to have new abilities. And some of the pioneer work that's done in the area started out 20 years ago to sort of engineer bacteria to have ability to oscillate a genetic circuit. So a protein is made, but it can have this oscillatory behavior. I think the field was really propelled by that ability. That first demonstration, and then since then, the field has grown a lot. And in part that's due to our ability to sequence DNA and be able to synthesize them. And we can program cells to do a variety of things. On the genetics part, you can have like I already mentioned, oscillator, toggle switch, and different feedback systems. But this is focusing on engineering native living organisms, oftentimes bacteria or yeast, and now more so with mammalian cells. So what is really the key of synthetic biology is the ability to have precise control over an input-output relationship. So if you think about a cell, the cells sends information, external information, and they're able to take that information and do sort of a computation and provide an output. So that's what a cell could do. And synthetic biology try to engineer that relationship, this input-output relationship. So where does synthetic cell fit in, right? So my expertise and my interest is in building synthetic cells, and that belongs to a subfield within synthetic biology that many of us call bottom-up synthetic biology, just trying to build things from the bottom up. Okay, so this is what my lab has been working on for the past 8 or 9 years now. And the premise of this field, or this bottom-up synthetic biology, is very similar to what I talked about just now, the synthetic biology, where we're trying to engineer cell-like system that, again, have defined input-output relationship, right? So a cell is if you're talking about animal cells, are usually about 10 to 15 micron in size, okay? Your hair, just to give you a sense of scale, is about 100 micron in diameter. So this is a fraction of the diameter of your hair. So a cell basically has a compartment, right? So otherwise it would just be a solution. But if we can engineer the cell-like system and allow the system to sense external stimuli and basically be able to program the response that it may have, then we have something like that is similar to a cell, right? So that essentially is the premise of the work, and then we call these synthetic cells because they're not natural cells; they're basically completely artificial. But we do use biological components in building these synthetic cells, right? So anything that we use are natural in the sense that they're biological molecules. And one of the defining I would say motivation behind this bottom-up synthetic biology is something you may have heard at the Ideas Lab that we talk about is the famous physicist Richard Feynman. He once said, what I cannot create, I do not understand. Okay, so that statement is very profound. Is essentially saying that if I cannot engineer a system, I just don't understand how it works, right? So we, for those of us that work in this area, we think building a synthetic cell is the ultimate way of understanding biology because we're building it. Okay, so if we can build a synthetic cell that can recapitulate certain life-like function, that will help us understand how cells, which are the building blocks of life, how they really work. And I think this is also the reason why NSF had a vested interest in trying to work in this area, because understanding the fundamentals of living systems, and this is a growing area and it fits really well within the convergence research framework that we just talked about earlier, right? Because it's drawing from a variety of fields into this into building a synthetic cell. So I hope that was reasonably clear for you on sort of
Can I ask a couple questions? And I don't know, well, they might not be useful on their own, but I wonder if it'll lead us somewhere. So if you're building a synthetic cell, just by way of scorekeeping, just by way of criteria, how do you know you've done it or you're closer? Is like self-assembly or replication something, or is it survives among other cells and doesn't get eaten? These are like, how do you know you're hitting the criteria?
Yeah, that's a great question. And I think this is the question that many of us are still let's say, struggle with or debate with among ourselves as to what constitute a truly functional synthetic cell, right? So, if you go to a origin life meeting where people are always argue about what is the beginning of life, and the question there becomes, if you build a synthetic cell, how do you know it's alive? And I think that kind of question is very philosophical and may not have a true one size fit all question answer. One would argue, okay, if you have a synthetic cell, we need it to replicate information, we need to have it divide, we need to have it grow. I think what we're trying to do, at least from my standpoint, we're not aiming for any one of these, right? To have those defining feature of life. Part of the reason for that is I think it's beyond our current reach. To be honest, right? So to be able to have something, and people do work on having a synthetic cell to grow, although the growth rate is really really slow, right? And then a lot of people are trying to make them divide, and this is a holy grail in the field as well. But I think to have all those features together in a single entity is somewhat challenging right now. So I think the other way to look at it is if you can define a particular, um, sort of cell-like function that you may want to understand better. And I think synthetic cell was a great approach to do that because we essentially strip down all the rest of the complexity and distill that into what are the minimal components that can reconstitute, that can recreate this process in a cell-like system. I think that may be more attainable. And I think this is where I feel like we'll have a much better way of justifying using this as an experimental approach. One of the things I also mentioned from time to time is biomedical research have been founded on the use of cells as model system, right? So we don't study, even though we want to understand humans or organism, we don't always study that particular organism, but instead we come out with model organism or model system. And cells have been a model system for biology for many, many years. So I will argue, can we use synthetic cells as a model system, right? By building that system, then we can start to add things in, take things out, and truly understand the mechanism behind how things work. And I think that is where, synthetic cells have a value. Does that answer your question?
Yeah, absolutely. Yeah. So do you want to tell us about building synthetic neurons?
Yeah, absolutely. Absolutely. I was going to just pause for a minute, make sure,
Yeah, no, perfect.
The synthetic cells are great. So yeah, so let me spend a bit of time to share with you, our Ideas Live project and sort of where that motivation comes in, and I'll try to tie it to the Ideas Live activity that we have. So when I think about building a synthetic neuron was a single idea that came up. One of the main ideas I suppose I brought up at the Ideas Lab, and it did bring together a number of investigators at the meeting, and we ended up writing a proposal that was funded. So it was a very I would say a great experience for all of us. So how does it come about? So I guess the context I like to share is that when I think about a synthetic cell, you can ask what cells do you want to build, right? So what there are many cells, and what type of cells do you want to build? And to put that in context, you and I, as an average human, we have about 30 trillion cells. Okay, so that is a pretty large number. And just for fun, our current national debt is about $27 trillion. Right? So it's on a comparable range. I often joke, if I can sell a cell for $1, then I can probably pay off the national debt.
Does that include microbiome, or doesn't include microbiome?
No, it doesn't include microbiome. It just be the cells that we have. Yeah, actually, it's interesting. Yeah, if we include the microbiome, that we could probably. It
goes way up. Yeah,
exactly. Yeah. Well, maybe it does. No, it doesn't. It doesn't. I know it doesn't, but I think microbiome is another. I don't know. Adds another. A few orders of magnitude, right? Yeah. So this is. The point is that this is a very large number. But nevertheless, there are only about 200 cell types in our body. Okay, so out of that roughly 30 trillion cells, there are only about 200 cell types in our body. And each of us, we all came from a single cell, a fertilized egg to begin with. So that fertilized egg has all the genetic blueprint, right, that gives rise to the myriad of cell types that we have in our body plan. And to me, that is just remarkable. So how does that single, cell with one set of DNA, give rise to all these, cell types? So I'm not saying development per se, or not really addressing that question, but to me, it's a fundamentally remarkable fact. Now, at the Ideas Lab, I decided to, I proposed to build a neuron. Out of that 200 cells, obviously there are many, many interesting cell types. I thought neurons was interesting. And why is that? And, so our brain obviously is a very important organ. There are roughly about 100 billion neurons in our brain. And the ability for these neurons to connect and function are what gives us the ability to think, to remember, to carry out our simple daily tasks. And everything that we do revolve around our brain functioning. So the fundamental question that is interesting to me is, how do cells communicate, and how do neurons communicate? So neurons are specialized type of cells, terminally differentiated. So once you have the neurons, they die, they're basically gone. And there's very little regeneration that happens. But the neurons themselves actually communicate in a very interesting fashion, right? So they communicate electrically. So in a membrane, there's actually charges that are separated by the lipid bilayer, which is the main component of a plasma membrane. And in the resting neuron, we have a negative potential of minus 70 millivolt. And the way that neuron communicate is through these things called action potential, electrical propagation that basically triggers from one neuron. It propagates down axon, which is a long, slender part of the cell, and then basically gives out neurotransmitter that then goes to the next neuron and then fires. The term we call is firing, and basically that connection or that the neuron fires the next one, and so on and so forth. So basically, to communicate using this sort of way of communication. And why is that important? So why, so how do we understand this process? So basically, at the molecular level, imagine you have a I'm sort of going to drop with my hand here. You have the left hand side. I'm just going to simplify the neuron into a cylinder. On the left hand side, you have basically in the resting neuron, basically the potential is all the same. But then we need to propagate this information across. And the cells uses ion channels. So these are proteins that are on the membrane that it allows ion to cross through. So in a normal cell, basically you have this minus 70 potential that basically we have a lot of sodium on the outside. And then we need to first elicit sodium ion to enter the cells. So basically there is a ligand-gated ion channel, and that ligand is the neurotransmitter. The neurotransmitter binds to the receptor, the ion channel, and allows sodium to enter the cell. So that basically raises this membrane potential a little bit. Okay, so we start from this minus 70. Once it raises to minus 50, then we have channels that are gated by voltage, right? So they're basically open. The open and closing state depends on the voltage level. So at about minus 50, another channel opens up, and this channel actually allows potassium to leave to essentially leave the cell. So the sodium continue to come in until you go up to the peak, and then after that, the potassium channel kicks in, and then potassium leaves the cell. So then you can get the negative potential back again. So you get this very characteristic action potential spike. So that's essentially the general concept of electrical communication by the neurons. So what we want to do in this synthetic cell is to basically replicate this. One of the goals is to replicate this idea of action potential, right? So if we can reconstitute, if we can put in this ion channels into our synthetic cells, and basically the hypothesis that we can recapitulate this entire action potential, because then we can have these ions that just come in and out, and be able to propagate this electrical information. So this is at a very, very high level what building the synthetic cell from the bottom up requires is the
So if I've understood correctly, it's trying to create a chemically triggered electric slingshot.
That's right. That's right. So so there are a couple feature I didn't quite go into depth, but essentially that's it. That's how the natural neuron work. Is they use chemical information, convert that into electrical information, and convert back to chemical information, because the neurotransmitters are the small molecules that eventually triggers the next neuron. So so I think I can start to put things slightly more into perspective. Our project now, so we start with the chemical information that then, like you said, is electrical slingshot that basically propagate that electrically because they're faster, right? The reason why we end up using electricity is they're faster than having chemical diffusion along a long axon. And then that electrical information is then converted to a chemical information again by, the last stage of that transmission uses a voltage gated calcium channel that allows calcium to come in, trigger the release of the neurotransmitter. But we're interested in actually all three aspects of taking the chemical information, propagating that to electrical, and then going back to chemical. But the first sort of challenge is to reconstitute these different ion channels. And instead of also using the chemical information to excite our neuron, we are also thinking of using light. There's been a lot of progress in using light to trigger neurons, living neurons, and these are called optogenetics. So we're also thinking of, in fact, working on using these light-sensitive protein that can actually regulate ion movement as a way to trigger this action potential. So we're doing both, light and chemical. So the work, so I'm kind of with that kind of background, I'm just going to quickly talk about, our little progress to date. So we had the project for about a year or so. So the project is quite large in the sense that there are 7 labs altogether that work on this. And I'll only, for now, just focusing on the science part. So my lab, I'm sort of the main PI on the project, but work very closely with the rest of the lab. So my lab has been using, using the technique that my lab developed, which is to reconstitute membrane proteins within synthetic cells. So we're reconstituting different channels. So the voltage-gated sodium channel, the voltage-gated potassium channel, and the inward rectifying potassium channel. Those 3 are the minimum channel we think will be needed to do the action potential, and we still need one more channel to excite it. So we're doing that part.
So in the lab, you're building membranes with sodium channels in them?
That's right. Yeah. So the technology is actually pretty straightforward. So and we've been working on this for a number of years now. We made vesicles, right? So a cell has to be a compartment, and we're the way we do this is almost like blowing bubbles. And basically the lipid bilayer is the material that that forms a cell, and basically we use a cell-free expression system. So this is a lysate that is extrapolated from cells that can undergo transcription translation, and basically we encapsulate that within this lipid bilayer, and these this lysate system can make membrane proteins and deliver them to the membrane. Some of the exact mechanisms of how exactly do that is unclear. We're actually studying that as part of another project, but the idea is that we can deliver membrane proteins into the membrane, and we're trying to do this with multiple ion channels. And the idea being that the ion channel is what really drives the action potential. So we have basically imagine the vesicle, a synthetic cell that has all these membrane proteins embedded within the membrane, and if we excite them, we can actually see. The hope is that we'll be able to see electrical activity within these synthetic cell. So hopefully I was clear on for that. And so I'm just going to mention a couple some of the progress on from the other lab. So we work very closely with Cindy Tang at Stanford. So her lab, she's a microphysicist. So essentially, I keep mentioning that neuron is a long cylinder, but really when we make these synthetic neurons, they're a sphere. They're basically a circular or a spherical cell. So they've been developing techniques to basically, right now to inject, using microfluidics, materials within these synthetic cells. So we call this pickle injector because the volume is very small. They're basically injecting small volume within this into the vesicle, and then they're also working on trying to deform these vesicle, hopefully into a very slender, more like a neuron shape synthetic cell. So those are the things that Cindy's lab is working on. And I keep saying we're building things from the bottom up, but I also need to broaden this up a little bit more. When we think about building synthetic cell, we're not just doing things from the very bottom up. So we also have a couple, team members. This is Changling Yuan from Purdue and TJ Ha from Johns Hopkins, and basically they're trying to do, working in living cells, try to engineer a protein system for a synthetic transmission. Okay, so this is in living cells now that we're talking about sort of a different approach to synthetic biology, still very much synthetic cell. So we're trying to basically create a new way for cells to communicate. So think about what I talk about the neuron. The way they communicate is through these synthetic transmission that involves a neurotransmitter and basically activating ion channels. But we're trying to do something that's different. We're trying to have them be excited by light. And the way this, again, conceptually, the way it works is we're engineering two cells. So imagine the two cells that one is a presynthetic neuron, one is a postsynthetic neuron, but these are not neurons, right? So we want to take normal cells and make them like a neuron. So the idea is that we'll put in, first of all, there are proteins that we're going to engineer onto both side of the cells, and we need to bring them very close together. And because the way neuron works is a very, very small, synaptic cleft, very small distance. So basically, we're building a synthetic cleft. Okay, so by bringing the two cells very close together, and this is using engineered proteins. But once they're close together, idea is that one cell membrane will have a bioluminescent generating protein. So bioluminescent is like things you will find in some certain sea organism that basically have output lights, right? So we'll have bioluminescence, that basically yield light on the one neuron, and then the other side will have this light activated channel. Again, those are engineered channel. They're basically called channel rhodopsin, and we can basically have the light that gives out from one cell. The cell basically emits light, and that will basically excite the postsynaptic neuron. So that's the idea that we're trying to work on, with Chun-Li and TJ. Very exciting ideas, and we don't know exactly how it works, but we're working on that. Very quickly, we have a computational biologist. This is Mo Das from Rochester Institute of Technology, and she is using the Hodgkin-Huxley model, and this is a mathematical model that describes how action potential neurons are initiated and propagated. They're using this base model to understand the phase space within action potential. So, within this parameter space, what are the parameters that really will give rise to a neuron firing? And we're able to recruit, and this is someone you haven't met, Francois Saint-Pierre at Baylor College of Medicine. So at the Ideas Lab, we didn't have a neurobiologist or neuroscientist in our team. We ended up recruiting Francois Saint-Pierre to our team, and he, he's in neuroscience, and he is using a non-excitable cell and then trying to understand how action potential can be created, by just introducing different ion channels, right? So again, coming back to the premise that excitable cells, a neuron is an excitable cell by electrical activity. It's really just the reason why it can be excitable is because of these ion channels. So if you take a non-excitable cell, okay, a cell that is on your skin, for instance, we can make them excitable by introducing different ion channels. So he is essentially exploring, what are the basis of action potential and how that action potential may change depending on the different amount, different type of ion channel we put in or different amount that we put in. So that's sort of give you a very quick status update on our synthetic neuron project, and hopefully that was good.
Yeah, very cool. It sounds, I mean, I could ask questions forever about all the details of how it works. If the thing that comes to mind is the idea that form follows function. Yeah. And so if you'll entertain one question, I'm wondering how you create shapes. So if you have a vesicle, which is just spherical in its nature, that's its most comfortable shape. How do you change shape?
This is also things that we work on in my other aspect of the lab, but which deal with cells. But Vesicles are these really fascinating material, right? So yes, you have a particular area, surface area, and you have a volume. You can osmotically deflate them. So in order to change shape, what you would do is to drive the water out. So you have an increase in surface area to volume ratio. So you can do that by putting it in a hyperosmotic condition. So it becomes floppy. So it's not quite slender yet. So they become floppy. And then what you could do
Like a
is you can
grape,
float
like a grape turning
like
into
a
a
grape.
raisin, so to speak.
Exactly. Yeah. So basically, you drive out the water, but obviously the in the raisin kind, when you dry them, they become crumbled, right? So they don't really maintain the shape. But the lipid bilayer is always a fluid, and you can
Okay.
maintain that fluidity of the membrane, but now they change shape because they're no longer spherical, and now you can shape them almost like like my daughter was playing with clay there. You can shape the clay. Obviously, clay is still a solid material, but this is something now you can then push it through a microfluidic channel, and you can essentially confine that vesicle into a cylindrical or a square channel that makes it thin, right? So you can start to do things like that, and and other people have done this, right? So if you take a vesicle that is spherical, you can osmotically deflate them and then push it through, push it through a channel, right?
Yeah. So you're working with pressure change, basically internal and external pressure changes.
Exactly. Exactly. Yeah. Yep.
Very cool.
Yeah.
All right. So, what are some of the impacts and implications, right? So, assuming you succeed
Yeah.
in building synthetic neurons, what does that mean?
Yeah, so I'll talk about impacts in a couple of ways. I sort of yeah. So one, I didn't quite cover all the team members' progress, and as you know, Barbara Hawthorne from UC Santa Barbara was one member of our team, and her work was geared towards, I'll probably start with that first before going to sort of the broader scientific impact. So her lab, as she's a social scientist, and really has brought a lot of interesting element to our team. And she's currently working on survey design and survey design development. And the hope is to understand how the public perceives synthetic cells. So when you talk about impact, one of the things that I think is important to know is that the impact, if it's impact to the scientific body, we can talk about that. But there's obviously greater impact, hopefully, to society. And with that, we need to also understand how the public perceive these kind of type of research, right? Because it's not, I would say it's not that conventional. So what they've done so far is they've done some survey, which I'll talk a little bit about, and also carry out a study on non-governmental organizations, so NGOs, how their activities is shaping the use, development, and handling of synthetic biology technology. So not just focusing on synthetic cell, but synthetic biology in general. And they can speak a lot more about what they've done, sort of looking at different NGOs and what their missions are, and how how they might influence public perceptions. And just understand who is actually the bodies that are influencing public's view. So far they have carried out the public interviews, and to me, one of the interesting questions, we have monthly meetings with all the 17 members, all the 7 labs, and in the December meeting, they give us an update, and one of the questions that they ask these interviews, so they interview 41 participants. Okay, so these are real sort of public, these are people that are non-scientists, and they asked them, how useful or harmful would it be to use synthetic neuron for different purposes? Okay, so and I'll tell you, one of the purposes, obviously, we say, okay, what if you can use it to treat injured people, right? And everybody said it was great, that would be super useful. And then, by comparison, we say, okay, what if we use the synthetic neuron to create super intelligent entities? All right, so and out of that 41, about 2/3 of them said it would be harmful if we start taking these synthetic neurons into building, super intelligent entities. So I think, uh, I guess one thing that I take away from that is, for the same kind of things that you build, there's always useful applications and harmful applications, right? Even though our goal is just to build a synthetic neuron, you can use it for obviously positive things or perhaps negative things. So anyway, so when it comes to synthetic cells, close about half the participant expressed concerns that synthetic cell could change the course of evolution. So that was also a question we asked them, is whether if we were to build synthetic cell in general, and this kind of tied to the what you were asking earlier, what function would we try to replicate, and if we have the cells that start to evolve, right? So I think there will be a legitimate concern as to how are we changing the course of life, right? Anyway, so that was a few highlights of what I recall from Barbara's lab. And so in terms of impact, let me just sort of say the what NSF was started out with is this rules of life, and I think we covered this a little bit earlier, that I think building synthetic cell helps us inform the rules of life. Okay. And it tells us how biomolecules are organized together to provide features that mimic certain lifelike functions. And I think this is a fundamental question, right? So maybe not everybody in the world cares, but there will be enough people that care about these. How do things come about? And I think in the context of synthetic neurons, the impact is that it would be a phenomenal achievement if either we can create a bottom-up synthetic neuron that can recapitulate this action potential I talked about, which I think, if we're able to reconstitute different ion channels in the membrane, I think we have a realistic chance of demonstrating that, action potential. And it will be great achievement if we can develop an alternative synthetic transmission strategy in living cells, right? I talk about how bringing two cells close together using light to activate one cell, and then channel rhodopsin to trigger, the post-synthetic neuron. So if we can do that, that will also be amazing, because then we now can take cells that are normally not, communicating electrically, and all of a sudden we can make them communicate electrically, right? So both of these will have, I would say, pretty significant impact. Yeah.
Very cool. All right. So a couple questions left if you're up for it.
Sure. Yeah. That's good. Yeah.
First one's just a fun one. So for you, what is something that's really exciting that's coming from another field, from outside of your domain of expertise?
Wow. Yeah. This is tough. I mean, there are so many things that excite me. One, obviously I can go talk about a lot of the scientific advances that happened in the past, 5 to 10 years. One thing that's been a big hit in science in the last few years is this idea of spatially resolved transcriptomics. So essentially, we have genomics that you probably know from, it's been around for a while, and then we have this transcriptomics, and basic ideas of being able to map being able to know what are the genes that are being expressed in the tissue, and we have databases on that already. But now I think it's really exciting to be able to have the spatial information. So giving it, a tissue that we, know these genes are being, being activated, right? But there's still a heterogeneity, right? So every cell is going to be different. But now we have the spatial information. People are working on these and have techniques to do that now. So it's essentially, one approach is to combine imaging and genomics together. They're able to capture the spatial informations along with single cell sequencing. So really phenomenal. And I feel like, that we have, the single cell RNA-seq, which is already pretty amazing. But now if we have the spatial information, and one area that's just being applied is in the brain, right? So we have all these, 100 billion neuron in the human brain, but even if you take a mouse brain as a starting point, now we can know, where the cells are, right, and what are each cells expressing, so in terms of the transient level, and then this will be combined with their ability to conduct action potential, right? So if you combine all these, then we have a complete map of these information. So the spatial information and is key to many many things in science, right? It's just not activity, but where is it happening? So there's a lot of effort going on in that. And just sort of I guess what excites me, other than just sort of the basic sciences, I'm also really interested in thinking about very, very long in the future, right? So going to outer space, right? So traveling to planet like Mars. I think hopefully within my lifetime we can see, maybe you can do that in a couple months, and maybe for amount of dollar that I can actually afford. So I think I was reading that it may cost, let's say, about $500,000 to travel to Mars one day. So hopefully that will happen, and I hope that's a round trip ticket, not
Yeah, I was going to ask one way or round trip.
Yeah, but I mean, things like the science and technology have progressed so much that things that I think about as a kid, and a lot of this are happening, right? Including travel to outside of Earth, right?
Yeah, it's incredible to see what gets achieved in such a short period of time.
Exactly. And other areas like artificial intelligence, deep learning, sort of data analytics sides is huge. I mean, there's so much exciting things happening right now, and that feeds in not just technology, but just also in the basic science, because the data that we collect are so much now that we really do need to have new tools to handle these. So, yeah.
Sir, the work you and your group are doing is truly transformational. I want to say thank you, at least on behalf of our team here at KI, if not, all of the beneficiaries ultimately of this kind of work. And I know sometimes it's hard to see, like I'm, speaking now for the general public, it's hard to see where scientific discovery meets, real life. And it might take a long time, but the smartphone in your pocket doesn't work unless you understand quantum mechanics. And we started in earnest to try to understand the behavior of electrons. Right. At that level, in the 1920s?
Yeah.
So it takes some time for the scientific discovery to, for the rubber to meet the road.
Yep.
But it changes everything when it does. So truly transformational work. Thank you. I suppose just as a closing note, it might be really nice to see if there's anything you'd like to say to young aspiring scientists out there who happen to catch this podcast at some point.
Sure. Yeah. Well, first of all, thank you for this opportunity. I really appreciate the kind words, and like you said, at the cutting edge of science, sometimes you don't really know the impact until it's really there. So another good example is laser. When the physicists, town that created laser, that was not something you thought would, transform the world in the way that it does now, right? Of course, everything has lasers in there. So hopefully one day these synthetic cells will really be of use in some fashion. So as far as aspiring young scientists, and this is something I take, a great pride in, is I love to talk to young scientists and find ways to motivate them and inspire them. I think one thing that I come to realize a lot in my times training student is is to keep an open mind. It's really important. I think oftentimes we're sometimes you can get very focused on say solving a particular problem and not really seeing the big picture. And I say to my student, keep an open mind about how you approach problems, how you think about problems, because the solution that comes oftentimes requires you to step a little bit outside of your comfort zone, maybe even in talking to people, especially to I think the open-mindedness is is one of those things that are, again, hard to teach, and try to preach it as much as I can. And it's not just about hard work. It's not just about being smart. It really is a lot of times about having an open mind. And I would say, given what we talk about at the beginning on convergence research, I think, practice both convergent thinking as well as divergent thinking. We haven't said much about it, but the things that we did at the Ideas Lab, the content generation process, that was divergent thinking. That is absolutely imperative to do that in order to explore a wide and diverse and substantial solution space. To think about how we can, take information from outside of our area and then focus on converging problems. So yeah, again, I think the divergent thinking will be associated with creativity. There's both convergence and divergence are absolutely, I would say necessary for success in science.
Thank you so much. And to anyone who's listening, we wish you luck on your creative, innovative, and scientific endeavors, whatever they might be. Thanks, Alan. Thanks, everyone, for tuning in.
Thanks, Asa. It was fun. Thanks.
Yeah.